Hi and greeting. It is pleasure that the International Academic Awards nominated my work in the category of the Best Research Award. I'm Samaneh Rahamouz-Haghighi, a member of Department of Plant Production and Genetics of University of Zanjan and a researcher of Zanjan University of Medical Science in Iran. Now I'm going to give a brief introduction to my academic experience and the kinds of literature review articles that I contributed with other authors can be seen in the first slide. There is a lot of progress in molecular regulation of biosynthesis pathways of secondary metabolites. The efforts in this research are restricted by the lack of efficient protocols for genetic transformation. For successful plant transformation, an efficient protocol for in vitro tissue culture and plant regeneration is necessary. So we decided studies to introduce simple and valuable protocols for callus induction, in vitro shoot organogenesis and plant regeneration of these valuable medicinal plants. Several articles in this regard are shown in the following slides. After that, our research focused on inducing the transformed roots and hairy root-derived plantlets. We evaluated their medicinal compounds and applied elicitors to increasing the yield of secondary metabolites. Several articles in this regard are shown in the following slides. We also further presented a protocol for the evaluation of important medicinal compounds of acubin and catalpol in different parts of Plantago species, whose details are clear on the opposite slide. I were interested in conducting studies in the field of evaluating the biological activities of medicinal plants, including anti-cancer and antibacterial, and I have done several studies about these, which have been introduced in several slides. Most of these studies are about two well-known and prominent species, Plantago lanceolata and Plantago major in traditional medicine. It is a pleasure that the International Academic Awards nominated my work entitled Phytochemical Screening and Cytotoxicity Assessment of Plantago lanceolata root extracts on colorectal cancer cell lines and branchium larvae and determination of the median lethal dose in mice in the category of the Best Research Award. This study reported a good relationship between the in vitro and in vivo tests, and these tests are a useful tool for predicting and confirming toxicity in plant extracts. After that, we designed another study based on the previous work. In the previous study, the crude methanolic extract had shown good cytotoxic effects, and in this regard, the methanolic extract was fractionated and the cytotoxicity activities of extract were investigated. I have published several studies on the biological effects of Plantago species and identified their effective compounds, and in these plants, iridioid glycosides are very important, and the most important of them are acubin and catalpol compounds. I decided to publish a review article that would improve my knowledge of my findings, uh, as titled Pharmaceutical Properties and the Analytical Methods for Determination of Acubin and Catalpol Iridoid glycosides in Plantago species. Thank for your attention, and you can visit the articles to see more details.